Welcome back, dear students. Today we're gonna be talking about production of the formed elements. The lifespan of the formed elements is very brief. Although one type of leukocyte called memory cells can survive for years, most erythrocytes, leukocytes and platelets normally live only a few hours to a few weeks. Thus, the body must form new blood cells and platelets quickly and continuously. When you donate a unit of blood during a blood drive, approximately 475 milliliters or about 1 pint, your body typically replaces the donated plasma within 24 hours, but it takes about 4 to 6 weeks to replace the blood cells. This restricts the frequency with which donors can contribute their blood. The process by which this replacement occurs is called hemopoiesis or hematopoiesis. Sites of hemopoiesis. Prior to birth, hemopoiesis occurs in a number of tissues, beginning with the yolk sac of the developing embryo and continue in the fetal liver, spleen, lymphatic tissue, and eventually the red bone marrow. Following birth, most hemopoiesis occurs in the red marrow, a connective tissue within the spaces of spongy, cancellous bone tissue. In children, hemopoiesis can occur in the medullary cavity of long bones. In adults, the process is largely restricted to the cranial and pelvic bones, the vertebra, the sternum, and the proximal epiphysis of the femur and humerus. This process is referred to as extramedullary hemopoiesis, meaning hemopoiesis outside the medullary cavity of adult bones. When a disease such as bone cancer destroys the bone marrow, causing hemopoiesis to fail, extramedullary hemopoiesis may be initiated. Differentiation of formed elements from stem cell. All formed elements arise from stem cells of the red bone marrow. Recall that stem cells undergo mitosis plus cytokinases, cellular division, to give rise to new daughter cells. One of these remains a stem cell and the other differentiates into one of any number of diverse cell types. Throughout adulthood, the liver and spleen maintain their ability to generate the formed elements. Stem cells may be viewed as occupying a hierarchical system with some loss of the ability to diversity at each step. The totipotent, toti equals all, the totipotent stem cell gives rise to all cells of the human body. The next level is the pluripotent stem cell, which gives rise to multiple types of cell of the body and some of the supporting fetal membranes. Beneath this level, the mesenchymal cell is a stem cell that develops only into types of connective tissue, including fibrous connective tissue, bone, cartilage, and blood, but not epithelium, muscle, and nervous tissue. Differentiation of formed elements from stem cells. All formed elements arise from stem cells of the red bone marrow. Recall that stem cells undergo mitosis plus cytokinases, cellular division, to give rise to new daughter cells. One of these remains a stem cell and the other differentiates into one of any number of diverse cell types. Stem cells may be viewed as occupying a hierarchical system with some loss of the ability to diversity at each step. The totipotent stem cell is the zygote or fertilized egg. The totipotent, toti means all, totipotent stem cell gives rise to all cells of the human body. The next level is the pluripotent stem cell, which gives rise to multiple types of cells of the body and some of the supporting fetal membranes. Beneath this level, the mesenchymal cell is a stem cell that develops only into types of connective tissue, including fibrous connective, bone, cartilage, and blood, but not epithelium, muscle, and nervous tissue. One step lower on the hierarchy of stem cells is the hemopoietic stem cell or hemocytoblast. All of the formed elements of blood originate from this specific type of cell. Hemopoiesis begins when the hemopoietic stem cell is exposed to appropriate chemical stimuli collectively called hemopoietic growth factors, which prompt it to divide and differentiate. One daughter cell remains a hemopoietic stem cell, allowing hemopoiesis to continue. The other daughter cell becomes either of two types of more specialized stem cells. Lymphoid stem cells give rise to a class of leukocytes known as lymphocytes, which include the various T cells, B cells, and natural killer NK cells, all of which function is immunity. However, hemopoiesis of lymphocytes progresses somewhat differently from the process for the other formed elements. In brief, lymphoid stem cells quickly migrate from the bone marrow to lymphatic tissues, including the lymph nodes, spleen, and thymus, where their production and differentiation continues. B cells are so named since they mature in the bone marrow, while T cells mature in the thymus. Myeloid stem cells give rise to all the other formed elements, including the erythrocytes, megakaryocytes that produce platelets, and the myeloblast lineage that gives rise to monocytes and three forms of granular leukocytes: neutrophils, eosinophils, and basophils. Lymphoid and myeloid stem cells do not immediately divide into and differentiate into major formed elements. There are several intermediate stages of precursor cells, literally forerunner cells, many of which have the suffix blast. For instance, megakaryocytes are the precursors of megakaryocytes, and proerythroblasts become reticulocytes, which eject their nucleus and most other organelles, hemopoietic growth factors. Development from stem cells to precursor cells to mature cells is again initiated by hemopoietic growth factors. These include the following. First, erythropoietin, EPO, is a glycoprotein hormone secreted by the interstitial fibroblast cells of the kidneys in response to low oxygen levels. It prompts the production of erythrocytes. Some athletes use synthetic EPO as a performance-enhancing drug called blood doping to increase RBC counts and subsequently increase oxygen delivery to tissue throughout the body. EPO is a banned substance in most organized sports, but it's also used medically in the treatment of certain anemia, specifically those triggered by certain types of cancer and other disorders in which increased erythrocyte counts and oxygen levels are desirable. Thrombopoietin, another glycoprotein hormone, is produced by liver and kidneys. It triggers the development of megakaryocytes into platelets. Cytokines are glycoproteins secreted by a wide variety of cells, including red bone marrow, leukocytes, macrophages, fibroblasts, and endothelial cells. Next factor is colony-stimulating factors. They are glycoproteins that act locally as autocrine or paracrine factors. Some trigger the differentiation of myeloblasts into granular leukocytes, namely neutrophils, eosinophils, and basophils. These are referred to as granulocytes, CSFs. A different CSF induces the production of monocytes called monocyte CSFs. Interleukins are another class of cytokine signaling molecules important in hemopoiesis. They were initially thought to be secreted uniquely by leukocytes and to communicate only with other leukocytes, and were named accordingly, but are now known to be produced by a variety of cells, including bone marrow and endothelium. Researchers now suspect that interleukins may play other roles in body functioning, including differentiation and maturation of cells, producing immunity and inflammation. That's it for today, dear students. Good luck. See you later.